So our picture is based on three main concepts. So the first one is indirect measurement, which is basically using a given quantity to estimate other quantities on either a larger or a smaller scale. And then another aspect is how features at the nanoscale can affect features at a human scale. And then the last kind of concept that this picture addressed was the idea of constructive and destructive interference. So in the picture, you can see in the top left corner that there is a key where one fish equals 4 circles of fish food. And then you can see in each bath or pool, there are varying levels of fish food and fish. So in the upper bath, there are more fish, so it makes sense that there would be more fish food. And then in the bottom bath, there are less fish, so there are less fish food. And each bath is 1 million liters. And then on the left side, you can see that there are two kind of groups of fish, and then one group has both lines of fish swimming in like a similar wave pattern, which is successful, and then there is another group of fish where their wave patterns are kind of contrasting, and then they end up bumping into each other, which obviously isn't successful. And then in the bottom left corner, you can see a person holding a bottle of oil, and they're pouring this oil into a pool, and then there are arrows bouncing off the oil, which is representative of how light waves would bounce off the oil in this case.
Okay, so as Mackenzie mentioned, the first topic of this picture is indirect measurement. So in the simplest terms, indirect measurement is using things we know to figure out things we don't know. So in this picture, we see one fish correlates with 4 food, and in the 2 tubs, the artist established that the tubs are the same size, so we don't care about the volume of the water. But we see that in the first tub, there are a bunch of dots, way more dots than the second tub. So using indirect measurement and knowing how much food each fish eats, then we know in the first tub, there's way more fish than the second tub. And our second topic was, how does things at the nanoscale affect the way we perceive things on a human scale? So that would be this small drawing down here. So in our lab, we measured the layer we measured the height of a monolayer. So we used olive oil, which mainly consists of oleic acid. Oleic acid is an amphiphilic molecule, so it has a hydrophobic tail and a hydrophilic head. So imagine my hand is the surface of the water, then the hydrophilic head, the water-loving part, is in the water while the tail is sticking up, which is the hydrophobic part. So when we say monolayer, that means just a single film of these molecules are lined up on a solid surface. So in our experiment, we did 2 trials, one with olive oil diluted in ethanol, and the second one, olive oil just by itself. In this picture, the person pouring the oil is just pure oil. It wasn't diluted with ethanol. That's why when the person poured it, it formed this cover that seems shaded in, but really it forms a rainbow surface. It's iridescent. Since a drop of oil, technically, if it has a very large area of water, it can form a single monolayer, but since the tub we used was too small, the layers started overlapping. That's why it affected the way light passed through the layer, creating this rainbow effect. Otherwise, the main point of the lab was diluting it in ethanol to make the volume of the oil so small that even in our small tub of water, when we put 1 drop of olive oil in, it can still form this single layer, and it should be transparent. And the last topic was constructive and destructive interference, as shown by the path of the fish. And the second part of the lab we did was using a laser beam and optical grating. So we shine the laser beam through the grating, and the grating separated the laser beam into different waves. And so if you hold a piece of paper to the end of it, then you would see like a row of dots, like a bright spot, a dark spot, a bright spot, and a dark spot. So when the light is separated into waves, then there's constructive interference, which means waves, the height, the peak of the wave corresponds with another wave's peak. Therefore, it becomes a greater amplitude, which is demonstrated by the light, the parts where the laser light shows. And destructive interference is when the peak of the wave and the trough of the wave, they cancel each other out, which creates these black spots.
And we really liked this drawing, and we thought it illustrated the concepts very well. And one thing in particular is that you could very clearly see how the troughs and peaks line up for the constructive interference. But one thing that we thought maybe could change is that with light waves, for example, the different waves combine to increase the amplitude. So in this case, they could have maybe drawn a greater wave or something resulting from that. A limitation of this analogy is that fish themselves can't combine, but that's okay. We thought the destructive interference was very well illustrated as well, with the fish canceling each other out. And we also liked how the person standing at the pool is able to see the interactions at the nanoscale level on how we could see it with our visual eye, or visibly ourselves. But another limitation of this drawing is that because it's from like our point of view, we're not able to see the hydrophobic and hydrophilic interactions themselves. We can only see how it's forming a layer on top.
Yeah. Any questions?
Oh, thank you.
Thank you.
Okay, we are open for questions. Rita gets to ask the first question.
Okay, sure. So one of the things I was thinking about with this drawing is I wonder if there's a way to use the fish to actually show amphiphilic molecules and the monolayer if you use the fish in the food. Can you guys think of a way of doing that where you could get some type of alignment with using the same things that were already in the first part of the analogy?
I guess the fish would want to go to the food, so I guess they would all line up, like with their heads on the top and maybe tails on the bottom. So they, like if the food floats on the surface, then they'll all line up to get access to the food.
And so in that case, like the head or the mouth of the fish would be like hydrophilic because it's attracted to the fish food, which is like representative of the water, and then the bottom half of the fish would be hydrophobic.
So then it would literally kind of be a hydrophilic, head and tail sort of analogy.
Literally head and tail kind of analogy. So the in phase and out of phase here, it's very clear, but I think, okay, so phase and waves are not an easy concept because they're not a concrete thing. So sorry, I have it's a little picture and I can only half see it. So in terms of the Auna phase, is there a way you guys could think of to modify the Auna phase to make a physical picture of destructive interference?
Wait, could you?
So in destructive interference, what happens to the wave?
Well, it cancels out, so like if the wave is going like this, then they'll both just kind of like flatten out, and they...
So how could you maybe capture that in the fish?
Well, since we all know, like when fish die, they kind of just float in a straight line. So when the fish, they collide, they give each other concussions, they die, and then they kind of just float in a straight line.
They could also just disappear. They could actually cancel each other out.
Yeah.
So I should point out for the audience that this experiment was actually had two parts that were totally separate experiments that were both about measurement at the nanoscale. And so for people who tried to capture both aspects of them, they are actually fundamentally disconnected because they were two experiments. In the future, we should probably encourage people not to capture both halves of them because they are actually totally separate experiments. Okay, so other questions from the audience? Yes.
So I was a little unclear about your experiment with the diffraction grating and a laser. Were you using an actual diffraction grating and a regular laser for that?
Yes.
So when you described that, you were talking about how it broke up into different wavelengths, but since the laser is, two within experimental parameters, one wavelength, how does the diffraction grating work for one wavelength?
Oh, I guess I wasn't very clear. So the diffraction gradient, it's the grating. It's kind of like an optical or
It's like
And multiple people, yeah. Other people should answer these questions too.
yeah, so it's like a bunch of like little slits, and so diffraction is like defined as when like a light wave passes through either an optical or a slit, and it like causes the light to like bend sort of, and so the thing that we use, it was basically like it was a bunch of it was like a crisscross pattern, so it was a bunch of different slits, so it caused the laser to kind of diffract in several different directions.
But these gratings were like very, very small. And with this, we used the indirect measurement to determine that the spaces were like on the nanoscale small.
So I guess to put this in a much more understandable context, so you know how like over a bridge there's often those arcs and water flows. So when water enters those, so water at first is like this continuous waves, but when it enters the two slits, then they kind of break up to their own individual and it slowly gets larger. So that's why when you go to the other side of the bridge, you see like these points of intersection and they slowly cancel each other out until it goes back to being one big wave.
Oh, and I don't think we mentioned this, but after we shined the laser through, the constructive interference made like 3 dots, or like dots appear in a line. So the laser was like split very orderly because of the nanostructure.
And then what could you measure once you saw the dots?
We measured the distance between the 2 dots, and using the height that we held the grating on, then we could use trigonometry to find the distance between the gratings.
Okay. So another form of indirect measurement of finding a nanoscale dimension. Okay, any other questions? If not, can our artists stand up?